Hi, I'm Gary Bates. I am a professor of biology at Northwest Arkansas Community College. I am also the Agriculture, Food, and Life Science Program Coordinator, as well as having other administrative roles at NWAC. Today I am coming to you to speak with you about CBEC as well as AR Cure and what that it's actually done for my classes as well as for my research program. So I would like to first talk with you all about the classes and how they've been affected. I actually used AR Cure with the Upward Bound program with some high school students. I've actually worked with Upward Bound for a very long time, longer than I care to admit. And we had those high school students actually do the 3A assembly, synthetic biology, and they were able to design as well as construct plasmids. Pretty proud of them for being able to do that. Those plasmids that they actually created were handed off to college students in my research lab for them to utilize and go further with. I have done a cure in the plant biology course, and whenever that we did the cure in the plant biology course, the students got to do plant tissue culture, and as that we did the plant tissue culture, we were propagating heirloom crops as well as some plant lines that produced phytochemicals. That is actually important for my research. And that was pretty nice. Whenever that you're utilizing these cures and such, it's quite handy because you can have a classroom of students do something and then hand it off for another group of students to do other work on. The results of that cure was actually observed by microbiology students as well as students in other agriculture courses as well as the entire horticultural program where they were able to see that. And we'll talk a bit more about that as we go forward. I also have a undergraduate research lab. My lab is ran with my wife, Dr. Lachelle Bates. You'll actually hear from her as well. The main focus of our lab is oxidative stress and its effects on both plants as well as animals, specifically mammals. We do plant tissue culture like I mentioned before. We have some lines of plants that we're growing out because we're interested in those phytochemicals and how that they will actually have an effect on different cell lines. We're working with U87 cells right now, kind of seeing how that those come together. We also do some engineering aspects in our lab. We do some precision agriculture, some robotics, design, utilizing Raspberry Pis, 3D printing, some of the Adafruit branded products, those aspects. But that's really not what we're wanting to focus on and talk about. We're going to talk about what AR Cure has been able to do and CBEC, which is the cell culture with the CBEC, as well as the synthetic biology with the 3A assembly through the AR Cure. I want to share with you some pictures that's actually coming out of the lab and such. First thing that we have a image of a kale. This isn't a regular type of kale that you would most likely have on your salad. It's a special kale that we have growing in tissue culture, and we can actually see our explants growing there. Down at the bottom we actually see some of those high school students that I'd mentioned, and those high school students are actually working on their plasmid right at that moment. The next image that we see a gentleman who's actually working on a electrophoretic gel. He was actually one of my microbiology courses, and like I said, he didn't do a full cure with that, but there was some pieces that we used. He's actually loading a gel out of that, checking to see if that a certain bacteria is actually being present. That gentleman actually ended up, based on doing that piece of research, if you will, in my microbiology class, he ended up joining my research lab. Lots of potential in that young man there. The next picture that we see is going to be the callus being formed. That's the little purple brain looking things that we see there. We have multiple calyces. That's where we're adding the hormones in before that we can start our differentiation process. The next picture that we see is going to be a myriad of different bacteria that we're growing out to determine if those bacteria actually have the plasmids, if that we were successful in that. One of the very nice things about this 3A assembly is the ability to add multiple colored reporters, and then you can have your time to see if they actually were created. Very, very nice out of that, something that you would be very interested in. Up at the top picture, we see the group of folks that's standing in front of a garden. That garden is actually a sustainability garden at Northwest Arkansas Community College. That is a garden that I am in charge of. Those students actually are the students that did the plant biology cure. This is a non-majors class. Two of those individuals in that photo actually joined my research lab. They both have very promising futures as well. 3/4 of those students, again non-majors, went on to take either other biology courses or other agriculture courses. And whenever I was in discussions with them, because I asked questions such as what are your plans for the future and such, 3/4 of that class actually said they have more interest in science now, that increased their interest, and they were wanting to take other courses. They wanted to get other experiences like this. Again, a multitude of them ended up taking some of my agriculture courses, and realistically, if that you don't understand this, agriculture is just applied biology, realistically. I consider that a great win out of that all those students were very interested. ARCure and CBEC are wonderful. They have helped my research program. We have taken students, of course, to meetings, particularly INBRI, which is very good for them. ARCure and CBEC has helped a lot. It's a very good program, and I think that you will like it. Thank you very much for your time, and have a great day.